So in dihybrid cross is a cross of two different characters. Two characters are involved in this cross, and these two characters are inherited by two separate genes which are available in two different alleles. That means two characters, two alleles. Two characters, two characters. Just for other example, let's take character 1 and character 2. For character 1 there will be a dominant allele and there will be a recessive allele. Similarly for character 2 there will be a dominant allele and there will be a recessive allele. Right? Okay. And when two pairs of traits are combined in a hybrid, that means character 1 and character 2 are combined in a hybrid which possess both character 1 and character 2. These characters will segregate in an independent manner, which means character 1 won't depend on character 2 and character 2 won't depend on character 1. Genes that are located in different chromosomes assort independently during meiosis. As I told you already in last video class that the genes which are located, or alleles which are located in a chromosome, they assort independently with each other. For example, we have a gene for length. If we have a gene for a length and other gene for say other gene for some other character, then these two alleles will segregate independently, which means this set of alleles won't depend upon this one and this one won't depend upon, let's take character 1 and character 2. So for character 1 won't depend on character 2 and character 2 won't depend on character 1, and because of that there are many possible combinations that may occur in a gamete. So let's take example here. So here is given that we have a homozygous round seed yellow cotyledon and wrinkled seeds green cotyledon. So yellow is round is a dominant, round is dominant, yellow is dominant. They are two characters. Similarly here wrinkle is recessive and green is recessive, right? These are also two recessive characters, two recessive characters, right? So when they combine, right? So for example, we are getting character 1 from one parent and character second, the dominant character, homozygous dominant character from parent 1 and recessive character from parent 2, and when the hybrid is formed, the hybrid will possess both the characters which comes independently from one another. Okay, and when this hybrid will form a gamete, there will be separate, there will be different combination, right? For example, if you see, so R, Y and Y. So how many combination can be formed? Let's check it out. Okay, one, this character, this combination RY or RY, otherwise or both recessive. Right, now you can see that all these gametes possess both the characters and these characters are independent from one another. So there are chances that they will make a different combination. Okay, and because of this independent assortment, this lead to genetic diversity. For example, see, a parent can have 4 kids and each kids are different from one another. Why? Because of the independent assortment of different alleles from one another, right? Therefore each child is different from the other child. Okay, if an individual produces genetically dissimilar gamete, it is consequences of independent assortment, right? As I told you that different characters assembled in the different gamete is only because of the independent assortment. That means one character is not dependent upon the other character. Now, through independent assortment, the maternal paternal members of all pairs were distributed to gamete. So all possible chromosomal combinations were produced, leading to a genetic variation. Since there are different kinds of characters combined in gamete, for example, now we have a round and yellow, right? And here we have wrinkled and green. So let's take an example from a chromosome, homologous chromosome. Let's take a homologous chromosome. Okay, so so here is capsar, and here is small r and small r. Similarly, it's a yellow, and here it's green and green. Okay, so during independent assortment, that means like we have meiosis 1 and meiosis 2. So actually what happens? Okay, so from interphase, when they enter the cell cycle, so they enter the meiosis. So when they enter the meiosis, you see the chromosome will be like this. Here also it will like this, and then what happened? So they go for meiosis 1. At meiosis 1, the homologous chromosome gets separated. Right? Hope you remember. Gets separated. And what happened? Meiosis 2. Sister chromatids get separated. Isn't it? Hope you studied. Okay, so this was interphase, and then we have meiosis 1, and from here meiosis 2. And so when meiosis 2 happened, then we have a separation of sister chromatid, isn't it? We have a separation of sister chromatids, separation of sister chromatid. Okay, let's put the chromatid here, chromatid here, then a chromatid here, and a chromatid here. Similarly here, and here also. Right. So this will be one gamete, second gamete, third gamete, and fourth gamete. Similarly in female, one gamete, two gamete, three gamete, and fourth gamete. So one gamete, two gamete, three and four. Here also 1, 2, 3 and 4. So what will be here, because this is a pure form, so it will be RY, RY, and same as RY and RY, and here also each one will possess the small r, small y combination, all the one. Okay, and when they come for combination, okay, so what will happen? Different types of combination can occur, and because of which it's a huge genetic variation can form. So it says that in sexual reproducing plants and organisms, due to independent assortment, genetic variations takes place, which is important for evolution. Okay, so because of the changes in the genetic variation, new varieties, new variation comes, which leads to evolution. Okay, then law of segregation is concerned with allele of one gene, but law of independent assortment deals with the relationship between genes. That's important. Like last video class you saw law of segregation, law of, that can be for your 1 mark question. Law of segregation concerned with one gene. That means we are talking about monohybrid. Right, so when you study monohybrid, it explains about law of segregation. But law of independent assortment, explains by dihybrid cross, and dihybrid cross informs about the interrelationship between genes. Right, so remember that law of independent assortment implied by dihybrid cross, law of segregation is implied by monohybrid cross. Okay, so the crossing of two plants differing in two pairs of contrasting trait is called dihybrid cross. So as I told you that in dihybrid cross we cross two plants differing in two pairs of contrasting character. As I told you that the character 1 contrasting, right? So it may be dominant and recessive, and character 2 contrasting, so that's why dominant and recessive. So when we combine or we cross these two characters possessing plant, right, that will consider as a dihybrid cross. So two characters are considered at a time, right? Two characters, character 1 and character 2, two characters are considered at a time. So in book with the example is about the color and shape, color of the seed, right? So the book explains the color of the seed and shape of the seed. In book, character 1 is given the color of the seed, or let's take color of the seed, color of seed, and character 2 shape of cotyledon. The shape was shape of the Okay, let's do that. Color of the cotyledon, that was yellow and green, and shape of the seed was round and yellow. Okay, let's take it as a shape of seed, because in book is given as seed. So the shape of the seed is round and yellow, right? It's round and yellow, and cotyledon color is yellow and green. Okay, so these two are character, right? So character 1 and character 2, we can take it as a character 1. Character 1, let me take it as a character 1. Character 1, and this as a character 2. Right, in this character 2, the dominant is yellow, recessive is green, and character 1, the dominant is round and recessive is wrinkled. Right, I hope you understand. Okay, the pure breeding round yellow parents is represented by genotype RRYY, and pure breeding green wrinkled parent is represented by genotype RR. Okay, so parent 1, let's take the parent 1 and parent 2. So parent 1 is represented by caps R, caps Y, and parent 2 is represented by small r, small y, small r, and small y and small y. So it represented the round shape of seed and yellow color of cotyledon. Round seed, yellow cotyledon, and here it's wrinkled seed and green cotyledon. Right, so during gamete formation, the paired genes of a character assort out independently of the other pair. That means this pair, right, let me change the color. Okay, so this pair and this pair, that means the pair responsible for seed shape and the pair responsible for cotyledon color assort independently from one and another, right? So it will be like R and Y. Similarly, R and Y. Right? So they assort independently from. They assort independently from one and another. From one and another. Okay, clear? Okay, next. During the F1 fertilization, that means when the F1 generation is fertilized, self-fertilized, each zygote with an equal probability to receive one of the 4 combination. Okay, so when we cross these two, what we get? We get F1 generation in the form of like this, or when you rearrange, you can arrange like this, right? And if you cross the F1 generation again, what will happen? So each one will segregate in an independent manner, right? So it will be independently assorted. That's what the concept want to explain. So see, right? So we produce F1 generation with the combination of, it's a heterozygous dominant condition, right? It's a heterozygous dominant condition already given. Heterozygous dominant, right? And when you cross F1 with F1, right, this combination came out, right? So how it came out, do you have an idea? Okay, so basically what it want to say that if we have 1 2 3 4, right, and you want to make combination, this is box 1 and box 2. Let's try to understand. That's why I'm making like this. So how many combination you can form? You can form, you have to take only one from each box. Okay, so 1 3, then 1 4, and here 2 3 and 2 4. Combination 1, combination 2, combination 3, combination 4. Similarly they did R and Y. So different combination, R Y, then R Y, or then R Y, or right. So combination 1, combination 2, combination 3, and combination 4. Right, so you can see different combinations, 1 2 3 and 4, right? Dominant recessive, recessive dominant, recessive. Okay, so when you cross the F1 generation and F1 generation, right, this F1 generation and F1 generation, when you cross, now these gametes are independent. They, like, this combination can go with this, or with this, or with this, or with this. They are independent, right? So they can go with anyone, right? And same for each one of other. So that means you can see that how many different combination can be formed. Okay, so let's see. Okay, the same they show with the chromosome, like I showed you already. Okay, so when they cross, so we have like 4 gametes, 4 different types of combination, and from parent 1, and 4 different type of combination from parent 2. So the total combination is 16 combination, right? So 16 combination. So you can see that 16 combination form. So let's see that in F2 generation there are 16 combination form. Okay, so these 4 types of gametes of F1 dihybrid unite randomly, that I told you. So randomly they can just combine with anyone. Anyone can combine with anyone, right? So you can think about that. So this one can go with this. Let me change the color. Okay, so this one can go with this, or this one with this. Okay, let's do it here. That will be easy. Let me write one more time here. Right, okay, let's make it one more time here. So we have right, this is a 4 combination. Similarly, when parent 2 also, this 4 combination we make. Okay, so you can see that each one of the gamete is independent. Like this gamete has full independence to fertilize with this one, third one, or fourth one. Similarly, the second gamete is also having full independence to fertilize with this one, or the fourth one. Similarly, the third one is also having a full freedom to fertilize with first one, second one, third one, or fourth one. Similarly, the fourth gamete is also have independence to get fertilized with the first one, second one, third one, and fourth one. So there are total 16 combination is possible. Right? Okay, so let's see. Okay, so as I told you that last slide, I told you that the dihybrid cross and its result led to the Mendel to propose the second law of generalization. It's called Mendel's law of independent assortment, like that I told you, right? So each gamete is independent to combine with the other gamete, so they are independently assorted, right? Okay, so let's see. Okay, let's see that there are different combinations we have. I already showed you. And when they combine, so this is a Punnett square, right? This is a Punnett square and Punnett square. So Punnett square, right? Punnett's square, and in Punnett square, so we have put the female gametes one side and male gamete in other side. So let's see the different combination. Okay, when this one fertilized with this one, or this one with this one, or with this one, or with this one. So same thing did with the all, and we got different combination. So you can see the phenotype ratio is 9:3:3:3:1, right? So what is 9:3:3:1? So round and yellow, you can see it's 9. Then 3 round and green. See how many round and green are there? First let's see for round and yellow. Okay, it's 1, 2, 3, 4, 5, 6, 7, 8, and 9. Right, so round and yellow is 9. Then round and green. Okay, so it's 1, 2, round and green, and 3. Okay, then what you can see, yellow and wrinkled. Yellow and wrinkled is 1, 2, and 3. Then wrinkled green is just 1, only 1. Right, so it becomes 9:3:3:1. That will be the phenotype ratio.